Sweet. Okay, awesome. How do I move this? Okay, so thank you for coming to week 10 review. We're just going to go through all the material from this week and try to give you a little good summary with some practice questions from the problem set. All right. Okay, so the first thing we're going to talk about is DNA polymerase proofreading. So the error rate without proofreading is 1 in 10,000 to 100,000 nucleotides. But with proofreading, that jumps up to all the way to 10 million, which might not seem like a big difference, but it helps loads in keeping the DNA without mutations. And so proofreading, the function is done by either an additional domain in the protein or a subunit. And that goes back to unit 1 where we talked about proteins, domains, and subunits. But overall, it's the same function. It senses the mistake, and then it moves the incorrect nucleotide to the exonuclease active site. And as Laura mentioned in class, it's a 3' to 5' exonuclease activity. And here's a little drawing. And then, so here is an example of the hand. I think she's gone over this in class, or she will tomorrow. But as you can see, it's in the P site, which is when it's polymerizing. And if it senses an error, that 3' end goes to the E site where the exonuclease activity can happen and it can replace that nucleotide. And so if DNA polymerase were missing its proofreading function, what phenotype would you expect to see? And this might, I think this is a problem set question. So this, maybe you've answered it or not. Does anyone have any ideas? I can't see the chat.
Oh, no.
All right, we'll just go through it. So if it were missing its proofreading function, you'd expect to see higher rates of mutations, more mistakes during DNA replication, and then mismatch repair could catch several of the mistakes, but not all of them. So that's why you'd have a higher mutation rate. Oh no. Okay. And is there any questions? Okay.
All right. Hi, guys. So we are going to talk about damage and repair now. So our first mechanism of DNA repair is mismatch repair, MMR. And what that does is... Sorry. Okay. So when DNA polymerase proofreading misses an error, mismatch repair will go in and fix that, and then our error rate goes to like one mistake. Sorry, we have. Anyway, one. I apologize, guys. One mistake per per billion nucleotides, and it's really important in MMR that the cell is able to distinguish the new strand from the parent strand. So we're correcting the right one, right? So like if we have a mutation on our new strand, we don't want to go and change the parent strand because then we're going to end up with an actual mutation at both sites. So if you want, you can unmute your mic or like put it in the chat. Oh gosh. I don't know how this computer, I don't know how to see the chat.
Yeah, I can't see the chat. I think you hover over here and you click chat.
Okay.
But if we see a red
Yeah,
bar,
so if you guys want, you can put in the chat, but or unmute your mic, or we can just go over it. But if MMR mistakes the parent strand for the new strand, if it fails to distinguish between the two, how many daughters would have a mutation, or how many daughter strands would have a mutation, and then how many daughter strands would have a mutation if MMR just doesn't sense the mismatch? I'll give you a few seconds if you want to drop that in the chat, or you can put in IDK and go over it after. Oh, somebody said okay.
I think we have to unshare to see it.
Oh, why is that?
I don't know. Oh. Here, just click share. Wait, if we go here, love share, and then we can see the. It's weird.
Okay, so wow, sorry, we're having technical difficulties. So all the daughters unless the cell die. So yeah, it would be all of the daughters if
Unless the cell dies.
unless the cell dies. It would be all the daughters if we mistake the new strand and the parent strand. Because if the cell, when it's doing MMR, looks at the parent strand and says, oh, this is the new strand, this is where the mismatch mutation is, and then it changes the parent strand, then we're going to have two mutated strands because it corrected the wrong one. It changed it to match like the incorrect nucleotide on the new strand. But if MMR just doesn't recognize that there's a mismatch at all and it overlooks it, then only one of the daughters will have a mismatch because the parent strand will stay correct, and then or one of the parent strands will stay correct, and then the new strand will just keep its mutation, and then when it goes to replicate, it'll have that there still. So this is kind of just a summary of that. If there's no repair, then one daughter will have a mutation. If repair occurs on the correct strand, the newly synthesized strand, which would have the mutation on it, then that would fix the error and none of the daughters would have a mutation. But if it happened on the parent strand, then both would have a mutation because you're changing it to the incorrect nucleotide. So MMR is sensed by a protein or proteins, depending on the organism, and they sense lesions on the DNA. So lesions happen when there's like a structural distortion. If you have, say, like a G matched to an A, they're not supposed to bond like that. So there'll be a change in the structure that these are looking for. And different organisms identify the new strand differently. So in prokaryotes, your parent strand or your mature strands are hemimethylated, which means that those are, they have methyl groups on certain sequences, and the cell will be able to identify them as the old strand because the new strand hasn't had time to become methylated yet. So it'll say like, okay, if there are methyls on this GATC sequence, this is not the one that we're correcting, this is the parent strand. In eukaryotes, we're not fully sure, but the hypothesis is that the new strand has more nicks because of Okazaki fragments, so that might be how it recognizes the difference between the new and the parent. So when the lesion is sensed, the area from either like the methylated region or the nick, whatever identifies the strand as the new strand, that region all the way to the mismatch will be taken out, and helicase separates that region, and exonuclease removes it. Then we have, because single-stranded DNA doesn't want to be on its own, it breaks really easily, we have single-stranded binding protein that comes in and protects that parent with the gap where the mutated region was, and DNA polymerase is recruited to fill the gap. It uses sliding clamp to kind of make the synthesis more continuous, because on its own DNA polymerase would fall off and it wouldn't go as smoothly. And then DNA ligase seals that last nick between the 3' and 5', because DNA polymerase just kind of leaves it off and can't seal that last bond. And then in prokaryotes, the new strand will also be methylated once it's corrected. Okay, so if anybody has any questions on this, you can unmute your mic because we're having trouble looking at the chat. Okay, I'm gonna move on then. And
All right.
give it to Van.
Some common types of DNA damage. You might have seen these if you've watched the lecture videos, but if not, we'll go through them now and then you'll get another refresher. So we've got base-specific DNA damage, and we're thinking of depurinations and deaminations. Depurinations are a little bit more rare. They happen about 5,000 times per cell each day. And so it's just the spontaneous breakdown of a sugar-base covalent bond between a sugar and a purine base. And again, a purine base can be an alanine or a guanine. And so there's two figures. One of them on the right I like is a little bit better. It's colored and it shows exactly what gets taken off. And you can see in the products, the depurinated sugar, it just doesn't have its its sugar-based covalent bond has been broken, so there's no more of that base on it. Whereas in deamination happens about 100 times per cell each day. And it's the spontaneous oxidative reactions that turn the CN bond, so an amine group, into a CO bond, which is a carbonyl group. And that changes the overall amino acid from cytosine to uracil. It might not be a big difference for us, just changing an N to an O, but the cell recognizes it as a completely different thing. And so usually a cytosine binds with a guanine. And so if there's a double-stranded DNA and the cytosine's bound to guanine and it suddenly turns into a uracil, they're not going to find that bonding interaction. And so there's going to be a little kink. So that's the problem with deamination. And then finally, there's pyrimidine dimers, which happens when you're exposed to high quantities of UV light. Like if you get a sunburn, this is probably going to happen in your DNA and your skin. And so it's linkages between adjacent pyrimidines. And so it tends to be thymine and thymine dimers that form. And as you can see on the bottom right image, there's that kink in the DNA chain. And so this results in mutations, and it's really hard for the cell to replicate that DNA. And if you have any questions, please unmute your microphone. All right. Sorry, you had the longer sections.
That's all right. All right.
You're rocking it.
So I think this is the last little leg that we're going to go over. We're talking still about repair. So our first mechanism is base excision repair, BER, and that's used to repair depurination and deamination. So basically, when we have depurination, when we're removing either that A or G, that's going to create an A basic site, a site where there's just no nitrogenous base. But with deamination, we have a uracil instead of a cytosine. So we want to get to the point where we're starting at the same place and our deaminated region, it turns into an A basic site. So for deamination, we're going to identify the uracil and then cleave the bond between the sugar and the uracil, and that's going to create our A basic site. And then from there, it's going to be the same because we're having an A basic site either way. So for both of them, we have an endonuclease called AP endonuclease and phosphodiesterase. They are going to cleave the phosphodiester bonds on either side of the nucleotide. So then we have our A basic nucleotide can be removed, and sliding clamp, again, is going to hold DNA polymerase onto the site. DNA polymerase is going to fill it in, and that nick is going to be sealed by ligase. And here is just kind of a diagram of that happening. I'm going to probably go by that one pretty quick. Our next one is nucleotide excision repair, and this is this addresses larger distortions. So pyrimidine dimers, they would be repaired with NER. And it's a similar starting step. First, we're going to have an endonuclease, which figures out where our lesion is, and then it nicks the DNA on either side of the lesion, and helicase unzips that strand. So it's removed on both ends, and then it's separated or cut on both ends, and then separated by helicase. So that region is removed, and then we have sliding clamp and polymerase again, so it's kind of the same theme, and our nicks on either side of that area that's filled in are repaired by ligase. So if you want to unmute your mic, or we can go over it after, what type of cancer specifically, what part of the body, I guess, would be more likely if we had a loss of function in the NER mechanism? Okay, I give it away. So skin cancer would become a lot more likely if we couldn't do NER, because like Van said with the pyrimidine dimer formation, oftentimes that happens because of UV exposure or any high energy particle or wavelength exposure. And that's what happens to your skin cells when you're exposed to the sun. So if we couldn't repair that specific type of damage, then skin cancer becomes a lot more likely. So our last type of damage, we have two ways that we're going to repair it. It's double strand breaks. So on our strand of DNA, or on our molecule of DNA, I guess both strands are broken. And that kind of has the same cause as the pyrimidine dimers. So if we have any high energy input onto the DNA, then that could cause a double strand break. And also unrepaired lesions. So if any arm misses like a pyrimidine dimer, that can eventually cause them to break. And we really, really don't want this because first of all, like they could break in the middle of a gene. They could cause chromosomal abnormalities. We could end up with the cell undergoing apoptosis. We could have cancer. There are a lot of bad things that could happen from that. So we are going to repair that either with non-homologous end joining, which is more common in humans, and we'll talk about why, or homology-directed repair, which is more common in E. coli and yeast. So non-homologous end joining basically shaves off the blunt ends of the broken DNA and with exonucleases, and then just joins the remaining strands together with a protein complex. And usually the cell will use that if there's no complementary DNA strand to use to repair. So this is pretty efficient. Like we like it because we don't need a homologous strand. It can happen pretty fast, but because we're just shaving off those blunt ends and then putting it back together, we can end up with mutations, either deletions or insertions. So for homology-directed repair, it's a little bit more complicated. Actually, it's a lot more complicated, but you don't really need to know the specific mechanism. All you need to know is that a DNA template is available, so an identical strand to the one that's broken, and that strand will invade the broken region and give a complementary sequence to essentially like build off of a repair that break from. And it results in less errors. You don't lose any information that gets shaved off with your endonucleases. So this is preferable overall. If human cells can do this, they will. Usually they don't, because you need that other strand, identical strand to build off of. So if you guys want to shout out, do you have a guess why human cells are less likely to use HDR? It is like, it's a preferable mechanism, but we don't often use it. All right, so hopefully you've thought about that. So human cells at any given moment are less likely to be dividing, so they probably don't have an identical copy of the broken strand, whereas like E. coli or yeast, they're dividing more often. In human cells, you would have to catch it either in like G2 or M phase when you have replicated DNA. And also, human DNA has a lot more non-coding regions than yeast or bacteria. So the odds of you shaving down a region that contains a structural gene that might lead to a harmful mutation is a lot less likely. So yeah, unless you guys have any questions, that was our very fast review for this week.
Thursday should have more problem set type questions. I know we had a few throughout.
And hopefully quick mail will be working so we can like
yeah.
post it more normally. But unless you guys have any questions. All right, thanks for coming.
Thank you.
Bye.